In this video, we will discuss about the paper that did this CRISPR screening to find out the genes that can cause proliferation, differentiations, and cell migrations. So, in order to do their studies, they use the HL60 cells, and first they check the construct where they use the guide RNA against CD4. They can see that CD4 expressions goes down compared to the control, and this no sgRNAs shows the high CD4 expressions. So they use those vectors to create this CRISPR pool libraries, and then they inject it into this. They transduce this into this UHL60 expressing dCas9 crab, and after 7 days, they divided the cells for half the cells for differentiation, half the cells for proliferations for 6 days. 6 days for proliferations and 5 days for differentiations, and then measure the cells for different functional studies, that is chemokinesis and chemotaxis. And for this for the case of this chemotaxis, they change the gradient in this transfer assay, and then check the movement of the cells after some times, and then collect the cell populations that are migrated. And in the case of chemokinesis, they keep the same gradient in the whole this transfer assay. They keep the same gradient in the top and bottom of the transfer assays, and check the migrations of the cells and correct the cells that are migrated. And in the case of amyloid 3D, they seed the cells and use the collagen and fibrin matrix of 125 micrometer, and then allow the cells to migrate in the matrix, and then collect these cell populations and do the studies of what are the genes that cause these cell populations to migrate. So first, and here the results they have shown the fraction of the cells that are able to migrate. In the case of this 3D amyloid, around 25% of the cells are able to migrate, whereas for the case of after 9 hours, whereas for the case of chemokinesis, after 2 hours around 18% of the cells are able to migrate, and after 6 hours around 30% of the cells are able to migrate. But for this chemotaxis, the more cells are able to migrate, that is after 2 hours it is around 30%, and after 4 hours it is around 50% of the cells are able to migrate. And once they find out there what are the cells are able to migrate, then they did the volcano plots to find out what are the gene changes that are occurs in this proliferation cells, differentiation, and cell migrations. So in all the three cases, they can see that some of the genes that are down regulated. For the proliferation, they can see most of the genes are down regulated. For the differentiations, they see around almost approximately half of the genes are down regulated and half of the genes are up regulated. For the cell migrations, they can see the most of the genes are down regulated, but some are up regulated also. And thereafter, they check what are the genes that are common in all the cases, and the total number of genes are shown on the horizontal side that can be seen here, and the genes that are common in all the three different studies are shown on the right hand side. So this indicates that most of the genes are like unique to each other for the proliferations and differentiations, whereas for migrations a very few genes are unique to migrations itself. And so far this is the overall idea how they did their genome-wide CRISPR screening for proliferation, differentiation, migrations. We will discuss further results in our subsequent videos. And in this video, which just covered the first figure where they shows that how they did their studies, and for the cell proliferation, differentiation, and migration, they use the wild type cells, that is UHL60 expressing dCas9 crab cells, or UHL60 wild type cells as a control. From that they are able to find out the genes that are differentiated. And here the map is also shown here, that shows that these are the control populations, and above this dashed line are the targets that are either upregulated or downregulated. So that's all for this video. If you need to understand more about this experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.